Hello, AP Bio aspirants. So this is a Unit 3 review, and we are going to start with cellular energetics. So there are some subtopics, for which the first subtopic is enzymes. Enzymes are biological catalysts, because catalyst is a chemical that increases the rate of a chemical reaction without itself being consumed in the reaction. The enzymes are those catalysts which work only for the biological reactions. Substrates are called the reactants on which catalysts work. And enzymes catalyze biochemical reactions. They affect the rate but not the equilibrium constant of a chemical reaction. And they increase the rate of a chemical reaction in both directions, forward and reverse. When a substrate interacts with the enzyme, it is converted to product faster than it would have been in absence of enzyme. The new model, induced fit model, works better, which says that enzyme and substrate, the active side of the enzyme is the place where substrate binds, but while binding, they both show flexibility towards each other so that they can have a proper conformational fit. Along with enzymes, some helper molecules are also required, which are called cofactors and coenzymes. Cofactors and coenzymes are very important for the chemical reaction. Cofactors, for example, metal ions, magnesium, zinc, and coenzymes such as vitamin-derived organic compounds like NAD, FADH2, etc. Enzymes bind their substrates transiently but not permanently. The portion of an enzyme that binds substrate and carries out the actual catalysis is termed as the active site, also called the hydrophobic pocket. Now coming to the regulation part. Enzymes can be regulated by the concentration of substrate or the presence of an inhibitor. Inhibitor can also be called as allosteric inhibitor because it binds to the allosteric site. On the enzyme surface, the place where substrate binds is called active site, and other than active site, any pocket which is present on the enzyme is called allosteric site. If a molecule which is binding to this site is an inhibitor, it will inhibit the enzyme substrate interaction in this way. As you can see, it has changed the active site. So what exactly enzymes do? They help in lowering the activation energy barrier. Assume that this is substrate and it has to travel this long so that it can convert into product. This is an absence of enzyme. Now in presence of enzyme, enzymes will help in lowering this activation energy barrier by taking a shortcut. So enzymes bring the reactant, that is substrates, together, put them in the right position and stress the formation or breakdown of the right bonds. That's how they convert substrate to product in less time. The rate of an enzyme catalyzed reaction is affected by several factors: concentration of substrate, presence of inhibitors or activators, and the temperature and pH of the environment. As discussed earlier, inhibitors are molecules that bind to enzymes and decrease the rate of reaction. They can do this by binding to the active site of the enzyme and preventing substrate binding, or by altering the shape of the enzyme so it cannot bind to the substrate. The one which we discussed earlier was the second case. That means they were preventing substrate binding by altering the shape of the enzyme. Whereas activators, they are molecules that bind to enzymes and increase the rate of reaction. Whereas activators are molecules that bind to enzymes and increase the rate of reaction. They do this by increasing the stability of the enzyme substrate complex. The third factor, that is temperature and pH of the environment, also affect enzyme activity because enzymes require optimum temperature in which they interact with the substrate. Most enzymes have an optimal temperature and pH at which they function best. Inhibition can be of two types: competitive inhibition versus non-competitive inhibition. Competitive inhibition means where inhibitor and substrate, they both compete for the active site, and if inhibitor is bound, substrate won't be able to bind. Whereas non-competitive inhibition is the one where there is no direct competition between inhibitor and the substrate, but when inhibitor binds to the allosteric site, binding of inhibitor to the allosteric site changes the active site in a way that substrate can no more bind. This is indirect competition, also called non-competitive inhibitors. Next topic of Unit 3 is photosynthesis. Photosynthesis is a reaction or a process by which plants and phototrophs, they convert light energy into chemical energy. Carbon dioxide molecule from atmosphere which comes from respiration, combustion. It is fixed by the plants along with water, and in presence of sunlight, it is converted to carbohydrates. Plus, a byproduct as oxygen is released. The chemical energy is stored in the form of sugars, which are created from water and carbon dioxide. This pathway takes place in chloroplast through photosynthetic pigments such as chlorophyll A, chlorophyll B, keratin, and xanthophyll, depending upon the type of plant. This process is also used by algae to convert solar energy into chemical energy. Oxygen is liberated as a byproduct, and light is considered as major factor. Light is considered as major factor. In the light reaction of photosynthesis, when sunlight falls on the chlorophyll, the electrons get excited in the photosystem 2, that is at 680 nanometers. Electrons, when they get excited, they transfer to the electron carrier molecule, starting from plastoquinone to cytochrome C to plastocyanin, and finally they are delivered to photosystem 1. Photosystem 1, which has wavelength of 700 nanometers, where NADP+, which is in the reduced form, accepts the electron and gets converted into NADPH. Meanwhile, when the electrons are being transferred from one electron carrier to another, the proton molecules, they move out of the membrane. Because electrons cannot travel alone, and they are always present along with hydrogen. One hydrogen molecule contains one proton and one electron. So by default, we have to understand that whenever electrons are being carried from one molecule to another, protons are also moving, as they cannot move alone. So these protons, they keep on going out of the membrane. Because of their deposition outside the membrane, the levels of H plus molecule outside the membrane increases. So now there is more H plus outside and less H plus inside, which creates an electrochemical gradient. Electrical gradient because of the positive charge difference, more positive outside and less positive inside. And chemical gradient because of hydrogen species difference, more hydrogen outside, less hydrogen inside. Now, because of this what happens, these proton molecules, they take help from ATP synthase complex. This enzyme helps in H plus molecule so that they can enter inside back through chemiosmosis process, because obviously we have high concentration of H plus outside the membrane, so it will try to get in back. It will try to make the equilibrium with the inside H plus concentration. In return, ATP synthase asks for the proton motive force, the force which is present in these H plus molecules. Using the H plus force from these protons, ADP is converted to ATP in the F1 subunit of ATP synthase complex, and that's how from the light reaction, ATP and NADPH are synthesized. So the products of light reaction are ATP and NADPH. That means during the day, ATP and NADPH are synthesized. These ATP and NADPH are used during the night in Calvin cycle, also called dark cycle, and light independent reaction. It is also called carbon fixing reaction. So in the Calvin cycle, which is a light independent process, sugar molecules are formed from water and CO2 molecules. So the carbon dioxide from the atmosphere is fixed with the help of the enzyme Rubisco. This enzyme Rubisco helps in interacting the starting material of the Calvin cycle, that is ribulose bisphosphate. 3 molecules of ribulose bisphosphate interact with 3 molecules of carbon dioxide, which are fixed from the atmosphere. So because the ribulose bisphosphate is a 5-carbon compound, so it becomes 15 carbons in total. So we have ribulose bisphosphate, which is a 5-carbon compound. 3 molecules of ribulose bisphosphate form 15 carbons. 15 carbons interact with the help of Rubisco enzyme with 3 carbons of carbon dioxide, and together 15 plus 3, 18 carbons are formed. So the resulting product has 18 carbons. 6 molecules of 3-phosphoglycerate. 6 into 3, 18 carbons. 6 molecules of phosphoglycerate. Now these 6 molecules of 3-phosphoglycerate, with the help of NADPH and ATP from the light reactions, is oxidized and 6 molecules of glyceraldehyde 3-phosphate are formed. One molecule of glyceraldehyde 3-phosphate can form one glucose, whereas the rest 5 molecules of glyceraldehyde 3-phosphate, they go back in the cycle and help in regeneration of the starting material, that is ribulose bisphosphate. It's called ribulose bisphosphate, RUBP. Next, we have cellular respiration. So all organisms undergo the process of cellular respiration. This is a loop with the photosynthesis process. So the sugar molecule and oxygen, which are produced as a byproduct, as products of the photosynthesis reactions, they help in complete oxidation of glucose to form CO2 plus H2O. So this reaction breaks down a carbohydrate and converts the energy stored in that molecule into most basic cellular energy, ATP. The step 1 of cellular respiration is called glycolysis, in which glycolysis, breakdown of glucose molecule, takes place. A 6-carbon glucose molecule is converted to 2 pyruvate, that is 3-carbon compounds, plus 2 ATP and 2 NADH. This is the evolutionary conserved process. This can be understood from the point that glycolysis is independent of oxygen, doesn't require oxygen, or it can take place in anaerobic conditions also. When glycolysis takes place, very small amount of energy is released. Glucose molecule, conversion of glucose to pyruvate, is a process which is independent of oxygen, so we do not require mitochondria over here, and it can take place in any organism. Once pyruvate is formed, now this pyruvate has to enter Krebcycle, that is step 2 of cellular respiration. But before pyruvate can continue on into the mitochondria to enter the Krebcycle, pyruvate oxidation takes place, which means pyruvate is converted to acetyl CoA. Pyruvate to acetyl CoA conversion takes place. This pyruvate can also convert to lactate or alcohol, which is called fermentation reaction, which we will learn later. Lactate or alcohol, which is in absence of oxygen, also called anaerobic respiration or fermentation. Now coming back to the topic, step 2 of cellular respiration is Krebcycle. This acetyl CoA enters the Krebcycle, and this process results in creation of many NADH and FADH2 molecules, and these electron carriers will allow a lot of ATP production in the electron transport chain. Now you will think that how NADH and FADH2 will convert into ATP. So let me tell you, because ATP is the energy currency. So assume that ATP is US dollars, but NADH and FADH2 are foreign currencies. Now we need to convert these foreign currencies into our own currency, US dollars, so that we can use them inside our body. With the help of electron transport chain, we will be converting these NADH and FADH2 into ATP, so that they can be used by the body to do work. Step 3 is electron transport chain. Electron transport chains works in the same way as we have learned in the photosynthesis process, where the protons were produced, these protons were used up by the ATP synthase, and then the ATP synthase takes the proton motive force and converts ADP to ATP. But the only difference is the electron carrier molecules and the complex inside. So the complex inside the electron transport chain is, it is happening inside the mitochondria. There are 3-4 different complexes which are extracting electrons from NADH and FADH2, and the electrons from NADH and FADH2 are being transferred, and meanwhile protons, they are accumulated outside. Now these protons, they want to enter back inside with the help of ATP synthase complex, and thus they convert ADP to ATP, and that's how ATP is produced as a product of glucose oxidation. If oxygen is not there in the cell, then the pyruvate is going to go under fermentation reaction, so which is also called anaerobic respiration. Without oxygen, the Krebcycle and electron transport chain cannot take place, and because there is no final electron acceptor, so pyruvate can be converted to either ethanol or lactate. That's all for the video, and all the best for your exam. Thanks for watching.